Hi, I'm Jacob Krol. I'm from the University of Colorado, Denver on Schutt's Medical Campus, and I'm part of the JRavi lab. And today I'm going to be talking about a web app developed in our group called MolEvolvR. It stands for Molecular Evolution with R. And it's primarily used to characterize proteins on evolutionary scale using domain architectures and phylogenetic analysis. So just a really brief outline, I'm going to talk about why we developed MolEvolvR, some of the methods involved, briefly cover some of the applications and results, some publications, and some future directions that are pretty exciting. So in this really basic diagram, so on the left, if you have a protein of interest that you want to analyze, there are a lot of tools out there that you can use, like structural and functional analysis. And you could also look at the physical chemical properties. And what we want to do with MolEvolvR is sort of integrate these and look at this for all of the homologs of that protein. So on the right, you can imagine the highlighted diagram is the protein on the left. And then we compare the domain architectures for all of the homologous proteins for that. And this is just a very basic diagram of our methodology. So we just run our proteins through BLAST and then interproscan on all of those homologous proteins. And then we trace back the phylogenetic data using EUTILs and other taxon grabbing software. And so just to go into the methods a little bit more, we use the delta BLAST algorithm. And how this works is in the diagram, you start off with a query sequence and you match this against a database of profiles or position-specific matrices. And then you take the alignment of those matrices and you, in the second to the nth iteration, you use that position-specific matrix as your input, and then you search that against a sequence database, and you get your homologous sequences that way. So that makes it a domain a domain-sensitive homology search, which is perfect for what we're trying to do. So if you aren't familiar with interproscan, essentially what this is if you look at the diagram, the first column is sort of your computational feature, and then that's used to predict the second column, the biological feature, and then the third column, that group is the actual component software and databases that is all bundled together into the interproscan suite. And then, as I mentioned before, I don't have a figure for this, but we use NTRES to grab the taxon data using the accession numbers from the proteins input and gathered from the homology search. So the actual input data you bring in can be a full sequence, or you can bring in accession numbers. You can also bring in midway into the pipeline. So if you want to run your own homology search algorithm, you could do that and bring in your BLAST output. And you could also just come in with your interproscan output and just visualize your domain architectures. And so one limitation with this approach, looking at things on a very large evolutionary scale, is that you have to run a lot of computation, and we run this on a single server. So for instance, if you have a single sequence and you want to get 500 homologous sequences, you run delta BLAST once and you have to wait until that run is done to run your interproscan. That already takes about 500 minutes, if I'm being generous, at about one sequence, or 1 minute per sequence. And then if somebody submits 500 proteins from a reference proteome, then we have 500 for the delta BLAST, and then we get 500 back for each of those. And now we have about 250,000 interproscan runs all running on one machine. So this is something that we're looking at scaling very soon. And this is roughly space-time complexity of a function of the number of inputs times the number of hits. So be sensitive with the number of hits that you use, and we're working on figuring out the right amounts that'll also capture a wide range of the distant remote homologs while still allowing your job to finish in a reasonable amount of time. So here's an example output figure that you get from our app. And I should mention that all of this is interactive and queryable. It's built with RShiny, and you can filter and you can download all these plots. And here we have a domain network. And so what this is, the nodes are domains, and the radius of the domain is the frequency. This is for all of the homologs, the frequency that domain occurs. And then the edges are the architectures. And again, the edge weight or thickness is the frequency of the domain architecture. And here, similar to what I showed before, this is a visualization of the domain architectures. So the rows here are homologous sequences, and then the columns here are the member databases of InterProScan. So, and they're all labeled with, in that legend down there with colors that shows which domain is where. And you can also generate multiple sequence alignments along with the phylogenetic tree. And you can download all these. They should be publication quality and ready. And then here's an interesting one. It's an upset plot. So on the left histogram, you have the distribution of domains. And then the one at the top, you have the distribution of the domain architectures. And then finally, to kind of reiterate, you can filter all of these using a queryable table. And so this will be reflected downstream in all the plots that you generate. So if you were to filter that third column, the species, for only humans, all of your plots would only include human sequences, and you can be more complex with these. You see the last column, you can kind of see how we store these domain architectures in text. They're delimited by plus signs, and you can use that and only filter for the domain architectures you want, which is pretty nice. And you can also add or remove columns. We have all of the member databases supported from InterProScan, so things like motifs, families, and that sort of stuff. But we only include the one here. You can just click the add or remove columns and add many more. So a little bit about just the underlying software that was used to develop MolEvolvR. It's built with R-Shiny, which kind of couples the front end and back end together. And then we wrap the InterProScan and Blast with shell scripts on our server. And we have some custom HTML, JavaScript, CSS to kind of make things look a little bit better. Tested on all operating systems and main browsers. And yeah, so the entire analysis, I should mention, this is a job submission site. So you send in your data and you will get a code and you come back and retrieve it later. And that'll all run in our server. But when the visualizations actually render, when you go to get your results, that'll be on the client side. And so one thing we're planning to do with this that might be exciting for more computational audience is that we want to give programmatic access to our plots written in R. And so what this would allow you to do is you come in with your data already prepared, and then you use our functions to just generate these plots, and you could use them in your own software. And I think a good way that you could test this out would be to just run it in the app, see what you want to get from your input, and then you can automate that with our R package into whatever software you'd like. And so some future additions, we want to add genomic context, the bottom left portion, number 3. And what that is so we were looking at only domains within a single protein before. But what if we look at the neighboring genes of that protein and check those domain architectures to sort of zoom out even more and get more insight on the genes of the organism, not just the single protein. So sort of what I mentioned before, we also want to support nucleotide sequence submissions. That's something that we get a lot. And also we'd like to make this more of like an engaging community type of thing, where we want to make our GitHub repo public, but we still haven't done that yet. We have a few keys in there from the past, previous students. We need to clean some of that out. But yeah, we'd like people to raise issues, give tutorials at conferences, and just keep adding onto the app. So some of the applications, a lot of them have been in microbiology. We have used this app to look at sulfur acquisition systems in Staphylococcus, for instance, and DNA helicase loaders in bacteria. But there's many more other applications that have gone on before I started working on the application. And I'd just like to thank my group, specifically my PI, Janani Ravi, and the software engineering team They've helped us migrate the entire web server from Michigan State University over to Colorado University, which was not an easy task. And I'd also like to thank, they're not on this slide here, some of the original developers of the app, Joe Burke, Lo Sozinski, and Samuel Chen. And if you'd like to check out the app, here is the URL. And yeah, that's it.
Some question? Oh, but I will start it for one question. I'm not sure to understand where the connection with the phylogeny in your. Maybe I missed something.
So I would say what I mean by phylogeny is just like using the homology search. So if you isolate a protein in its environment, you only get to see that single protein. But if you look at the homologous domains of that protein, you can see how that domain has been spread out over time, over the tree of life. So that is sort of what I mean.
Okay. Thank you. Yes, Aida?
Thank you for your presentation. So you talked about the wanting to engage. So are there automatic tools to help people to contribute to the tool?
Well, I would say a good place to start is setting up like GitHub issue templates. That's a very easy way to start, and just having a forum on GitHub. We do a lot of back and forth with collaborators through email at this point, so we definitely need to find a better way. I think having a private GitHub repo makes it very difficult at the moment, but that was our main plan, is just to do most of it through GitHub and outreach events like this where we get to talk to people about it.